Hi, welcome to Myers ASMR. I thought about you, and I'm so glad you're here. How are you doing today? You feeling good? Good. Do you know that well-being dominates? Yes. The well-being of the planet is what dominates. In spite of whatever might be going on, everything's all right. Well-being dominates. So, let's get into today's story time. Super interesting. I found this at Strand Bookstore in New York. That is a wonderful place. I'm so glad bookstores still exist, especially bookstores like that. I think they have 3 levels and they're still thriving. Yeah, it's a wonderful thing that people still like to read books. So this is the Handbook of Human Sexuality. It's an old book. That's okay. Okay, so part 1, they talk about developmental phases, prenatal influences, and subsequent sexuality. It's typed like almost in a scientific way. So I have a feeling this is going to be some heavy duty stuff. Look how it's typed. But I got it because it still looked interesting. Terminology and definitions. Gender identity or role is a relatively new term in sexology. Obviously, sex and gender overlap, but the two are not synonymous. Gender encompasses sex, but conceptually is more all-inclusive. That is to say, gender extends beyond genital sex to include those aspects of male and female dimorphism behavior included that do not pertain directly to the organs of generation and the erotic and procreative process itself. Thus, gender dimorphism applies to male or female differences in legal status, vocation, recreation, grooming, matters, and cosmetics, but it does not exclude sexual status, psychosexual status, sexuality, erotic practice, and erotic imagery. Gender identity or role signifies the unity of gender identity and gender role. According to Money, 1965, gender identity and gender role are defined as follows. Gender identity is the sameness, unity, and persistence of one's individuality as male, female, or ambivalent in greater or lesser degree, especially as it is experienced in self-awareness and behavior. Gender identity is the private experience of gender role, and gender role is the public expression of gender identity. Gender role is everything that a person says and does to indicate to others or to the self the degree that one is either male or female or ambivalent. It includes but is not restricted to sexual arousal and response. Gender role is the public expression of gender identity, and gender identity is the private experience of gender role. The differentiation and development of gender identity or role is best conceptualized as a program that involves diverse variables interacting sequentially during critical periods of prenatal and postnatal development. X and Y chromosomes. The program of gender identity or role commences with the sex chromosomes. Nature's plan is that the X or Y sex chromosome of the male parent, when added to the one X sex chromosome invariably supplied by the female parent, determines the genetic sex of the offspring. Should a Y chromosome be added, the chromosomal pattern is XY, and provided prenatal development goes according to plan, male differentiation occurs. Should an X chromosome be added, the chromosomal pattern is XX, and provided all goes according to plan, female differentiation occurs. Nature's regular plan for the X and Y chromosomes is not always the one carried out. Among human beings, the known chromosomal anomalies include the loss of either the X or Y chromosome, the addition of one or the combination of more than one chromosomal pattern in the same individual. The term for this latter type of genetic anomaly is mosaicism. When mosaicism occurs, one or more supernumerary chromosomes may be present, or a chromosome may be missing in some cells but not in others. An example of the latter type of mosaicism is the 45x46XX pattern found in some girls with Turner syndrome. Concerning chromosomal loss, it is possible for one of the X chromosomes from the XX pair or for the Y from the XY pair to be lost without lethal effect. When either occurs, the result is a phenotypic female minus fertility with a 45X chromosomal pattern, Turner's syndrome. Girls with Turner syndrome have gonadal streaks in the place of ovaries. Consequently, hormonal replacement is necessary for them to have a feminizing puberty. Psychosexually, they are assigned and reared as girls, and they develop a stereotypically feminine gender identity or role. By contrast, the loss of an X chromosome from the XY combination is lethal, whereas the addition of one or more X or Y chromosomes is not. Examples of two chromosomal patterns with an extra sex chromosome are 47XXY, Klinefelter syndrome, and the 47XYY. Individuals with Klinefelter syndrome are morphologic males who have a small penis. The testes are small and sterile. Such individuals are greatly susceptible to severe mental retardation or psychopathology, which may be of almost any type, including the sexual psychopathology of transsexualism or transvestitism. The occurrence of psychopathology and mental retardation is sporadic and not a consistent concomitant of the supernumerary X chromosome. In morphologic phenotype 47XYY, individuals are male. They are usually tall, many over 6 feet. Sterility is not uncommon. Money and Associates compared 47XYY individuals and 47XXY individuals for behavioral disability, sexuality, and social interaction. Those 47XYY individuals with behavioral disability often were found to be characterized by impulsive acting out, example, destruction of property, and poor long-term planning. By contrast, 47XXY individuals were often found to be characterized by deficiency or inhibition of action, example, phobia. With regard to sexuality, the 47XYY individuals showed a diversity of sexual experience, whereas the 47XXY individuals were sexually rather inert or homosexual. Socially, both 47XYY individuals and 47XXY individuals preferred being alone to being with a group. There also exists a 47XXX condition, which occurs with a female morphology and a female gender identity or role. Fertility may or may not be diminished in this condition, and there may or may not be behavioral disability. HY antigen. The Y chromosome programs the differentiation of the undifferentiated cells of the primitive gonads into testes beginning at around the 6th week of gestation. Differentiation of the undifferentiated gonads into ovaries does not begin until the 12th week and requires the presence of 2 X chromosomes and no Y. The Y chromosome programs the undifferentiated gonads toward testicular development via a plasma membrane protein, the Y-linked histocompatibility, HY antigen. Exactly how the undifferentiated gonads in XX individuals are programmed into ovaries are not clear. Ono suggests the possibility of an ovarian organizing antigen similar to that of the HY antigen. This ovarian organizing antigen has not yet been identified. Having programmed the differentiation of the gonads, the sex chromosomes have no other known direct influence on subsequent sexual behavior and psychosocial gender identity or role differentiation. The program of gender identity or role differentiation and development is now carried forward by the presence or absence of secretions of the newly differentiated fetal testes. Without the secretions of the testes, morphologic differentiation is female. Adam-Eve principle. Prenatally, the program of gender identity or role is carried forward specifically by the determinants of masculine morphologic differentiation, namely Mullerian inhibiting substance and androgen secreted by the testes. Present knowledge of embryology indicates that fetal ovarian hormones are not essential to female morphologic differentiation. Whether or not maternal and placental hormones are essential is not known. Nature is predisposed first to make a female, and only with the addition of testicular secretions does it make a male. The embryological fact that nature's preference is female is epitomized as the Eve principle. The fact that something must be added to make a male is epitomized as the Adam principle. Morphologic differentiation of the genitalia. The internal genitalia differentiate ahead of the external genitalia. Prenatal hormones, their presence or absence, control the differentiation of both. The male and female internal genital structures differentiate from 2 separate primordia, the Wolffian and Mullerian ducts, respectively. In normal male development, the fetal testes first secrete their 2 hormones, androgen and Mullerian inhibiting substance, MIS, at around the 6th week of gestation. Androgen ensures that the Wolffian ducts proliferate into the vas deferens, seminal vesicles, and the ejaculatory ducts of the male internal genitalia. In the absence of MIS, the Mullerian ducts proliferate into a uterus, fallopian tubes, and the upper vagina of the female internal genitalia. In normal female development, the fetal ovaries do not, so far as is known, secrete hormones prenatally, and if maternal or placental hormones play a role in female differentiation, it has yet to be demonstrated. Whereas the male and female internal genital structures differentiate from 2 separate primordia, The external genitalia develop from the same primordia. These structures remain undifferentiated until approximately the 8th week of fetal life, at which time androgen secreted from the fetal testes begins to masculinize them. It is possible that testosterone is converted to 5-dihydrotestosterone, DHT, before masculinization of the external genitalia is effected. In any event, in the presence of androgen, the genital tubercle differentiates into a penis. The skin that becomes the hood of the clitoris and labia minora in the female wraps around the penis to cover it and to form the foreskin. The labial scrotal swellings fuse and form the scrotum. In the absence of androgen, the genital tubercle remains small and becomes the clitoris. What in the male is the foreskin and outer covering of the penis becomes the hood of the clitoris and the labia minora. In the female, the labial scrotal swellings remain separate and form the labia majora. Fetal androgen programs not only the differentiation of the internal and external genitalia, but also the differentiation of sex-related pathways in the brain. Exactly how androgen affects brain differentiation is not known. It is probable that it acts on neural substrates, which in turn regulate thresholds for the expression of sexually dimorphic behavior. Cyclic secretion of gonadotropins is sexually dimorphic. Hormonally, females cycle and males do not. In female rats, the ventromedial and preoptic areas of the hypothalamus regulate the cyclic secretions of gonadotropins. If female rat fetuses or neonates receive androgen, then in adulthood they are acyclic. Well, that completes today's story time. Like and subscribe, donate to my Cash App. Catch me in the next video, and may you find lasting happiness. Bye.